Hey guys. So this should be Tuesday, and at this point you should have already picked up all of your papers. So let's talk about what's happening today and kind of assign some things and move some stuff around on your calendar. Okay, so today's agenda, we will talk about and discuss, and I will assign the evolution take-home test, which does count as a full-blown test. It's in that big category. So hint, hint. We'll do a little bit of a genetics recap that should get you thinking about some words and some terms that you saw freshman year, or at least whenever you took on level biology. And then we're going to do some Hardy-Weinberg practice, and it'll come in the form of we'll practice the theory a little bit, and then we will practice the actual calculations and the interpretation of those numbers. So your focus for today is to be aware of all of the changes and to adjust your own calendar as I do so through the video, to review the genetic terms, like I said, from freshman year. And then we're going to look at some questions, one of which will be an FRQ practice question. So you're going to learn when to use the actual calculations, like the actual equation, versus when to use the theory or the rules behind Hardy-Weinberg. And then lastly, I'll start you off on the calculation and interpretation of Hardy-Weinberg, and you'll have some time to practice. We will be going the entire time. I would suggest that you take out a sheet of paper so that you can write some notes for the genetics recap just off to the side for yourself. So go ahead and grab your sheet of paper and then move the take-home test packet that you picked up to the front. Also, tonight you should be watching your phylogeny video, so you should have your note sheets for phylogenies as well. All right, so find the image that looks like the one that I have on the screen. This is the AP Biology Evolution Take-Home Test. It is not the same thing as those 4 essays I gave you. Those are separate. Those have a different due date, okay? So this take-home test is due, let's see, when is it due? October 6th. Okay, the take-home test is due on October 6th. Now, it's not as daunting as it looks. It is 14 questions total, but all of the questions are multiple choice. If you skim through real quick, you'll notice all of them have answer choices. Now, they also have lines, not a lot of lines, but some lines nonetheless. So let's talk about what it is you're doing. If you look at the front of the packet, you should notice that some of the questions have a little asterisk next to it. So like question 1, here's my asterisk. Then for question 2, there is no asterisk, okay? So with the ones that have the little star or asterisk next to them, you must do those, okay? You must do the questions with the little star next to them. The other questions that don't have the star, you have to choose 5 of those to do. So you're doing all the questions with the star and 5 of any other question that you choose. I don't care which questions, just as long as you pick 5. This is going to be the part that you must pay attention to be able to get full credit. You can't just tell me what the right answer is. Okay, so you have to circle the right letter choice, and you have to explain why that letter choice is correct, but you also have to explain why the other 4 choices are incorrect. Your book would be a good resource for this. You can kind of go back to some of our videos for this. You can do some research on your own, and you can always come ask me about questions that you're not super sure of. Some of these things we've already covered in notes. Some of them we are getting to. Okay, so again, not due until October. Actually, it should be October 5th, not 6th. My bad. Pretty sure the 6th is a Saturday. Yeah, so it's due October 5th. Okay, so hopefully this makes sense and you've had a chance to adjust your calendar. Feel free to write it on here as well. And then you can put that away. If you would please pull for me your hand lab. The hand lab is the lab packet that I gave you last class where you had to calculate the mean and the standard deviation and all of those. And you were supposed to draw 2 graphs for me. So go ahead and pull that. Okay, if they need a second to find their lab, you can pause the video at this point. So, let me adjust this real quick. I asked you to have your graph drawn. Graph 1 and graph 2, I believe, is what I asked you to do for me. So your graph 1, and I'm gonna do a rough sketch, should be, your histogram, however that worked out, whatever that looks like, that should be your graph 1. And then your graph 2 should be the normal distribution, which is what you would get if you kind of sketched the outside curve. Sorry. Okay. Windows really wants me to know that they're making it better. Anyway, if you sketch the outside curve of where those columns fall, that's what your normal distribution should look like. Okay. And all of this was based on the data from your table, the data with those numbers that I'd already given you. Okay, so what you need to do on your graph 2 is you're going to draw in, and you can use colors or you can shade in like different patterns or whatever, your standard deviation for graph 2. So on graph 2, you would take that normal distribution curve that you figured out, whatever yours looks like, and you are going to plot your standard deviations 1, 2, and 3, plus or minus 1, 2, and 3 within, based on that data. And then you are going to color or shade in the different parts of the standard deviation curve. Okay, so remember that's your 68%, your 95%, and your 99.7%. Okay, so that's what your graph 2 should look like. Now, graph 3 is going to be a replica of graph 2 in the sense that you're still doing a normal distribution curve. So for graph 3 and 4, you are going to replicate graph 2. Oops. Okay, so pretty much take your graph 2 and draw the exact same graph on spot 3 and spot 4. You should be writing this down. Okay, if you have time at the end of class, then this would be a good time to work on that. Okay, now you'll be using your graph 2, 3, and 4 to do part 3 of the lab. So once you've finished graph 2, you've finished part 2 of the lab. Okay, so in part 3 of the lab, you're looking at what's known as modes of natural selection. What would happen if this scenario were to play out? How would that affect where my frequencies and my numbers fall? So what they do is they give you, we're on page 12 of your handout, they give you some general information, excuse me, about the organism that your hand length represents. In this case, you guys are birds, so your hand becomes your beak. So they tell you things like each bird only has one beak, none of the birds can use tools. If you don't pick up food, then you will die, things like that. So read through that information. Then if you skip on down to page 13, it gives you what you need to do on each of the graphs coming up. So the first one is a directional selection like scenario. So it tells you a fungus infects all of the plants that produce smaller seeds. Most of these plants die. Based on scenario A, draw a new curve on graph 2 that best represents what will happen to the population under these conditions. And then you're supposed to answer the analysis and conclusion questions, which we will talk about. So let's talk about what that looks like. So you had this graph, and we know that our smaller numbers were down here, and our larger numbers up here, which means that our medium or middle numbers are in the middle. Okay. Now the scenario and our distribution looks like this. It's a normal distribution, meaning that our extremes have the smallest frequency. So we have a few birds that have really small beaks, a few birds that have really large beaks, and nearly everybody is in the middle. Okay. The majority of your population has a medium-sized beak, if you will. So the scenario tells us that a fungus infects all of the plants that produce smaller seeds. So there are no small seeds being made, or there are less small seeds being made. Now it stands to reason that if your beak is small, you probably eat smaller seeds. Excuse me, sorry y'all. It also stands to reason that if your beak is larger, you eat larger seeds. If you have a really large beak, it might be harder for you to eat small seeds. Okay, so logically speaking, that's where we are. So if these trees or plants are no longer making small seeds, then most of those plants are going to die. So if a bird has a small beak and can only eat small seeds and its food source goes away, hopefully you're thinking along the same lines as I am, it means that those birds with smaller beaks are going to start to die off. You'll have less birds with small beaks. Oops. Okay, so the question's asking on this graph, what is that curve going to look like? Okay, how are we going to change where the curve falls? Will it fall in the same place? Will it move to the left or to the right? Will it get bigger, taller? What's going on? So if I don't have small birds, I probably shouldn't have too many organisms that exist in this range of my graph. So think of it as I'm now going to shift my normal. I'm going to shift the direction in which my graph is built. Now, as far as I know, nothing's happened to the plants that make large seeds. So I probably still have some medium-sized birds and some large-beaked birds. So it means that my distribution is going to shift that way. So if you will, when we draw it in, we would draw it like this. Okay. And all this is showing me is that my population has shifted in terms of where my frequencies fall. I now have almost no birds with small beaks, and I have way more birds with large beaks. So that's why it's called directional selection, because I've changed the direction of my frequency. Okay, then oops, sorry. Hold on 1 second. Okay, so I'm going to skip to a new page. Okay, so the next option is disruptive selection. So when you disrupt something, it's almost like you stop it. You stop whatever was currently happening from happening. So we go back to the original, which is I have small beaks, medium beaks, and large beaks. The little synopsis says the bird population is transported to another island. On this island, the predominant plants are ones that grow in the safety of crevices, which are cracks in tree stumps or rocks or soil, and ones that produce very large seeds. Based on scenario B, draw a new curve on graph 3 that would best represent what will happen to the population under these conditions. Then they want you to answer the analysis questions. So again, I had normal distribution. Most of my numbers are in the middle. My extremes have a smaller population. So disruptive selection is going to be one that eliminates or reduces where the max of your frequency falls. So the majority of our numbers fall in the area that I've just shaded. Disruptive selection is going to reduce the number or amount of organisms that fall in this middle range. So when you draw it in, it will look like this. We're now going to have two pockets, if you will. We'll have lots of birds that are small, that have small beaks, lots of birds that have large beaks, and the birds that have medium-sized beaks, we're going to have a very small amount of those. Oops, let me change colors. Okay, so that's my disruptive against disruptive because I've effectively crushed or reduced or removed where my largest numbers fall. Okay, and then stabilizing selection is the last one. So same thing, small numbers or small beaks, medium beaks, and large beaks. Still a normal distribution pattern. Yeah, well, you notice that the graphs are getting wonkier as we continue. So this scenario says a large predator is introduced to the original island where the bird population lives. The birds with the larger beaks are usually the ones with the largest biomass. Hold on 1 second. Okay, I'm back. Yeah, so we were talking about the fact that the birds with larger beaks are usually the ones with the largest biomass. This makes them the slowest birds in the population. You think of mass meaning, amount of matter in them. Excuse me. So this increases their risk of predation. So if you're a big bird and a slow bird and a heavy bird, you probably are a bird that doesn't fly very quickly. So the bigger, slower, and heavier you are, the more likely or easy it is for a predator to get you. So it goes on to say the predator's urine is highly acidic and is killing off most of the plants that grow in the crevices. Remember those plants that grow in the little cracks and roots and stuff. And those are also the ones that produce very small seeds. So I hope you notice what they just did. They gave us a reason that our large-beaked birds and our small-beaked birds are probably not going to be very successful. Our large-beaked birds are not going to be very successful because they are bigger and easier to hunt. Our small-beaked birds are not going to be very successful because their food source is again being destroyed through the predator's urine. So they're asking, what would stabilizing selection look like on a graph? Well, we're going to reduce greatly our two extremes, our small and our large. And we might increase the frequency of the medium size, but we're also going to narrow the frequency. So we're going to define what medium means a little bit more. So let's go this one down a little bit. This one's going to look like this. Oh, crap, sorry. Okay, so that one's stabilizing. Excuse me. Oh, sorry if I'm moving too quickly. Pause if you need to. Okay, so this is what the graphing is expected to look like. So now let's talk about what are you doing in terms of the questions. So you're not going to do any of the questions for part one because remember, we didn't do part one at all. Okay, so this is under the analysis. So you can completely ignore all of part one questions, which are on page 19. On page 20 are where the part two questions are, and I only need you to do the following. So you will do you will do 4, 5, 7, and 8 from part two. Okay, and then for part three, you will do 9, 10, and 11. And for the conclusion, so I wrote part 4, but it's actually just the conclusion. So for the conclusion questions, you're going to do questions 3 and 4. Okay, and that's it for your lab. Excuse me. Right, so don't forget that the hand lab is due on the 27th. Today is the 25th, so still plenty of time to adjust and get it done. Okay, so at this point you need to, let's see, let me check my checklist. So we talked about the take-home assignment. We've talked about the hand lab. Excuse me. Let's do our genetics recap. Okay, so you need a sheet of paper to take some notes to the side. To be honest, I'm going to couple some of the Hardy Weinberg stuff with the genetics recap. So, we're kind of hitting two birds with one stone. Okay, so we are now going to jump a little bit back and forth between some genetics review, and we'll talk a little bit about Hardy Weinberg equilibrium as well. So let's start off by coming up with a working definition for what the Hardy Weinberg principle or model is. Very often it's called Hardy Weinberg equilibrium. Some people refer to it as a principle or a theory. Some people will call it a law. All the same thing though. So Hardy Weinberg were actually two separate people. One was a statistician, the other was a physicist. They came up with this theory that in their minds proves that evolution doesn't take place. To be honest, what they ended up doing was giving us even more proof that evolution happens. It didn't really work out the way they anticipated that it will, or it would, I should say. So, Hardy-Weinberg principle states that allele and genotype frequencies in a population never change. They stay constant as long as you don't have other evolutionary influences. So, let's write that down. So, we're going to say that allele and genotype, and we'll talk about why we have to specify both. So, allele, pretty sure I spelled that wrong. Or at least it just looks wrong. Oh, whoops. So allele and genotype frequencies in a population remain constant. Okay. And remain constant in this situation doesn't mean, hey, for this week or this year. We're talking generation to generation. Okay, now the only way that this can really happen is if there's no other influence coming from an evolutionary perspective. Okay, so allele and genotype frequencies in a population remain constant from generation to generation in the absence, and that's a very important part, in the absence of evolutionary influences. Okay, so this is what the principle states or says. So, what possible evolutionary influences would change this? Well, there are 5 conditions or 5 principles. Okay, so you want to hear my life still? Wait, hold on. Hey. Oh, Jesus. Okay, thank you. Bye. Oh, sorry. Really thought I'd hit pause. This is what happens when you have to break up fights after school, or at least call to report about fights after school. Ugh. Okay, so I was saying 5 principles to the Hardy-Weinberg equilibrium. They're all based on evolution and influences or evolutionary influences. So you are going to need to know these. The first principle says that a population has to be large. You have to have a large breeding population, breeding in the sense of capable of producing viable offspring. Okay, so large breeding population. Hardy-Weinberg does not work if the population is small. Okay, you need it to be a big population on the off chance that, let's say, 10% of your population dies. If your population is really large, you're not going to notice the loss of 10%. If your population is really small, there are 10 individuals in your population, you are going to notice the loss of 10%. So that's what the large breeding population does. It accommodates for random losses. The second principle says that mating is a random process. Okay, so this means that everyone has the equal chance of being able to produce offspring. Okay, the females in the group or the males in the group are not looking for a specific set of criteria. They don't care what you look like, smell like, how big you are, anything like that. They just are, if you're healthy and you're of mating age, then you are able to mate and produce offspring. That's what random mating is. We're not looking for something specific. Okay, 3 is that we have no mutations because mutations create differences or changes or variation. And variation means that my frequencies are going to change. My phenotypes are going to change. There is no movement between populations, no immigration or emigration. Okay, another way they phrase this is they call it gene flow. So whenever you leave like a city or a town that you've lived in and you move to someplace new, your genes move into that population with you. Okay, and then the last condition is no natural selection. The environment does not create situations where some individuals are more successful and live for longer than others. All right, so those are your 5 conditions for Hardy-Weinberg. So Hardy-Weinberg questions are going to come in 2 formats. There's either going to be, here's the problem where you can use the equations, or it's going to be theory-based where you have to talk about one or more of these 5 conditions. Okay, so now that we've gone over the theory, let's start looking at some of, not exactly the equations yet, but we're going to build into that. So let's start by talking about allele frequency. So this should be review because you had, or 99% of you should have had biology prior to now. So you're going to have to pull in some of that prior memory. This is your first time for bio, then we're starting from the ground up. So let's start by talking about what an allele is. An allele is a version or a type of a gene. Okay, so if you think about Chips Ahoy cookies, there are about 14 different types of Chips Ahoy cookies in the supermarket right as we speak. But they're all Chips Ahoy cookies at the end of the day. However, each package is slightly different. Like you might have crunchy and chunky and chewy and reduced fat and the ones that have the Oreos stuffed in them and whatever other kinds you have. But at the end of the day, they're all types of Chips Ahoy cookies, but they're slightly different. So that's what an allele is. It is a version of a gene that you get. And you always, as humans, we always get 2 alleles. One allele comes from our mom, one allele comes from our dad. Okay, and you'll see me use these symbols. These symbols don't mean mom and dad, but this one does mean female, and this one means male. Okay, so let's do an example. Let's say that we have a gene for hair texture. And you have 2 options. You can have wavy hair, or you can have straight hair. I hope you guys appreciate my amazing drawing skills that don't exist. Okay, so those are my 2 alleles. I have an allele for wavy hair, and wavy hair is going to be dominant. And I have an allele for straight hair, and straight hair is going to be recessive. So let's write that down. So big W, we're going to use that to represent wavy. And, oh, sorry. and wavy is dominant. Oh, seriously. Okay, so like I said, wavy is dominant. And then we have little w, which is going to represent straight, and straight is recessive. Okay, remember dominant means that you will be expressed. You make a lot of that protein, so you're going to be expressed. And recessive means that you don't make as much of the protein, or maybe some of the protein is non-functional, so you're not as easily expressed. Now, in terms of your genotype, so in terms of these letters, there are two ways for you to have, there are three different genotypes, sorry. So you can be homozygous dominant, and you would be big W, big W. These are our genotypes. You can be heterozygous and be big W, little w. I don't know why I keep writing my W's all weird. And you can be homozygous recessive, where you're little w, little w. Okay, that's for our genotypes. There'll always be three of them. For your phenotypes, you just have two options, at least with this type of genetics. When we get to actual genetics, we'll talk about some exceptions to the rules. But for this type of genetics, you can be wavy haired or straight haired. Those are the only two options. Okay, to be wavy hair, you can have two different options in terms of your genes or in terms of how your alleles arrange themselves. You can be big W, which we already knew, but if you're big W and little w, you'll still have wavy hair. Because remember, that little w means that you don't make as much of the protein or you don't make the protein at all. That big W, because it's dominant, as long as you have one big W, that's what counts. That's going to be the phenotype that you have. Straight hair is a recessive trait, which means that you can only have straight hair if you have two little W's. That's the only way that works out. Okay, so let's look at an example. Because we haven't actually talked about the frequency part yet. So let's say we have two people. So person one has the following genes, and person two has the following genes. Okay, that's what the alleles look like. So big W, and little w, little w. Okay, it's a small population. It's just easier to work with these two people. So let's think of your frequencies as the number of times the individual allele is represented. Okay, so the number of times the individual allele is represented. It's going to be important that you remember that this is what we're talking about. Okay, so let's break down what our frequencies could be. So if we took the frequency of big W, what would the frequency of just that big W be? Now, you might be tempted to say, well, in this person, it's 1 out of 2, and in this person, it's 0 out of 2. But you're thinking of the number of times the individual allele, so the one big W, is represented total in the gene pool, total in the population. So maybe we should add that to our definition. In the population. Okay, so think of each letter being a representation of a chromosome, and that gene is on that chromosome. So now we don't have two individuals we're looking at. We're looking at in the 4 chromosomes we have in front of us, so 1 chromosome, 2 chromosomes, 3, and 4, how many times do we see the big W? Okay, so if you think about it like that, the result is going to be 1 out of 4. 1 out of the 4 is the same as 0.25, which is 25%. Okay, so now let's do the same thing with the little w. What's the frequency? How many times are you seeing the little w represented in the population? Let's go back and look at the population. Excuse me. So we have 1, 2, 3 times. So 3 out of 4 times this little w is represented in the population, which is the same as 0.75 or 75%. Okay, so we can say that the number of times our big W is represented, we can use a variable for that, and we'll call that variable P. We can use a variable to represent the number of times our little allele, so in this case our little w, is represented, and let's call that one Q. Okay, so if you notice, if you added P and Q together, even in this example, we would get 100%. And it's not because I designed it that way, it's because it'll always add up to 100%. So P plus Q equals 100%, or another way to think about it, if we're thinking decimal version, we could say that P plus Q equals 1. Right, so there we are for allele frequencies. Now, we can also talk about the phenotype frequencies. So remember, your phenotype is your physical trait. Or physical characteristic. It's what's expressed or shown. Okay. And for your phenotype frequency, you have to consider the number of times that trait is shown. So not the chromosomes, but just the trait, period. Remember, in this situation, the trait is wavy hair or straight hair. Okay, so this is the number of times the trait is shown in the population. Okay, so if we go back up to our people, this would be wavy hair. This one would be straight hair. Because we're thinking of the person or the individual and not just the alleles, our population is 2. So if we were to look at the frequency of, or the percentage of wavy haired people, only one of those people had wavy hair. So it would be 1 over 2, or 0.5, or 50%. All acceptable ways of writing this information. So now that we've talked a little bit about the allele frequency, let's talk about Hardy-Weinberg equilibrium. I'm not going to go over the theory again because we've already addressed that, but let's start looking at some of these variables and where our two equations came from. Okay, so we've already seen how P plus Q gives you 1 when P is the dominant allele and Q is the recessive allele. So let's take a little math break and see if we can make some of this make more sense. So we know that P plus Q equals 1. We square both sides. Squaring a polynomial or binomial. Sorry about that. Tired. The result is going to be P squared plus 2PQ plus Q squared also equals 1. Well, we know that P is the dominant allele, and we know that Q is the recessive allele. What do these other things mean? Where do they come from? What's their purpose? So let's start with P. P squared is the same as saying P times P. So another way to think about it is it's like asking yourself, what is the probability of getting two dominant alleles randomly? Okay. Well, two dominant alleles, if you have two dominant alleles, for example, big W and big W, that's just like being homozygous dominant. So really, P squared is the chances or the probability that you can be homozygous dominant for whatever that gene is. Then let's jump to Q squared because that follows a similar trend. Okay, so Q squared, if P squared is P times P, it stands to reason that Q squared is Q times Q. And if P squared is a probability of two dominant alleles, then Q squared is the probability of two recessive alleles. Okay, or the probability or chances of you being homozygous recessive. Okay, so that's so far so good. That's not too bad. But 2PQ, that one sounds tricky. So 2PQ, you have to think about it from two ends. You got to think about the fact that you have a parent A and a parent B, and you have two possible outcomes. You can get a dominant allele or a recessive allele easily from either of them. And again, this is all random. So 2PQ is the probability of getting a dominant allele from parent 1 and a recessive allele from parent 2. But it's also the probability of getting a dominant allele from parent 2 and a recessive allele from parent 1. Because either parent, assuming that they have both types of alleles, can easily give it to you. It's a 50-50 chance for the most part. I'm going to give you a minute to digest that. But what we've just done is we've discovered where our two, oh, by the way, this is the same as being heterozygote or heterozygous. So really what we've just done is we've just discovered our two Hardy-Weinberg equations. Equation 1 says P plus Q equals 1. Equation 2 says P squared plus 2PQ plus Q squared also equals 1. We use the first equation for allele frequency calculations, and we use the bottom equation for genotype, phenotype, and individuals. Okay, so at this point, we need to get into some practice. So you should have somewhere close to you in that stack that you picked up a sheet called Hardy-Weinberg problems. Please find your Hardy-Weinberg problems for me. Yeah, that's not what I wanted. Okay, so I'm going to walk you through the majority, if not all, of this sheet. And then I will assign you which problems I want out of the other packet for you to practice either in class or for homework. You will be tempted to want to do this however you feel like doing it. Some of you will try to do proportions, some of you will try to do all kinds of things. I promise you, if you follow the method I lay out, you are going to be okay. Okay, so starting with number 1, problem 1 says that 25% of a population is homozygous recessive or Q squared for a particular gene. Calculate the following, and then it gives you two things. Calculate the percentage of the population that's homozygous dominant and the percentage of the population that is heterozygous. Now, it's always tempting to want to start with P, mostly because it comes first in the formulas, so everybody feels like that's where we start. But you actually always want to start with Q squared. The only way that you are going to start with anything other than Q squared is if they give you numbers that represent P or Q. Not P squared or Q squared or 2PQ, but just P or Q. Okay? If you don't have P or Q, you need to start with Q squared. So we know that we have 25% of our population that is homozygous recessive. The reason we're starting with Q squared is because there is only one way you can be homozygous recessive. There are no tricks to being homozygous recessive. You have to have both small or lowercase alleles, both recessive alleles from your parents, one from mom, one from dad. So this is how I would set it up. Q squared is equal to 0.25. So from this, I can now find Q, because if Q squared is 0.25, it stands to reason that Q is going to be the square root of 0.25, which is 0.5. So far I found Q squared and Q, or I have Q squared and I've now found Q. If I know Q, I can find P, because P is going to be equal to 1 minus Q, which is 1, which is, Jesus, listen. Okay, so let's try this again. 1 minus 0.5. Okay, and that's also going to give us 0.5. So Q is 0.5 and P is 0.5. From here, I can now find 2PQ. 2PQ is literally 2 times P times Q, which in this case is 2 times 0.5 times 0.5, which will give me 0.5. So I've just found all variables to my Hardy-Weinberg equation. I know Q. I know Q squared. I know Q. I know P. And I know 2PQ. Now I could go ahead and find P squared, which would be 0.5 times 0.5, right? So in that first part of the question, that's actually exactly what they're asking me for. What is P squared? So let's go ahead and write in P squared. Okay, so if P is 0.5, then P squared is going to be 0.25. So now I have all parts of the Hardy-Weinberg equation. So let's go back to the question. The question says, what is the percentage of the population that is homozygous dominant? Well, the percentage of my population that is homozygous dominant is going to be 25%. You want to keep your answers specific to the way they are asked. If the question asks for percentage, then I need you to turn your decimals into percentages. So 0.25 or 25% would be the answer for number 1. I do want you to show all your work, though. If you just give me random answers and I have no clue how you got there, I'll never be able to help you figure out where you're making your mistakes. Okay, so let's look at 2. 2 wants to know the percentage of the population that is heterozygous, or 2PQ, for the same gene. Well, we've already found it. Okay, that was our 0.5. So 2 is 0.5, which is actually 50%. So in the little bit of time it took us to solve all 5 variables, we then had the answers for both part A and part B of question 1. If it were me, I would go through and always find all the parts, because it doesn't take any time, and then I would read the questions and plug them in from there. Okay, so we've done it's really question 1A and B. So let's look at 2. So in question 2A, they told us 30, or not even 2A, let's just look at 2 itself. So 36% of a population is homozygous recessive, or Q squared, for a particular gene. Calculate the following. Okay, so before we even look at the problems, let's just go do the entire thing. So, come on. Okay, Q squared is equal to 0.36, which stands to reason that Q is going to be equal to the square root of 0.36, which is 0.6. P will be equal to 1 minus 0.6, so 0.4, and P squared will be 0.4 times itself, or 0.16. 2PQ is going to be 2 times 0.4 times 0.6, which would give you 0.48. Okay, so we've solved the entire thing. All variables are accounted for. So now let's look at the questions. Part A wants you to calculate the percentage of the population that is homozygous dominant, or P squared, for the gene. So my P squared value would be this right here. So my answer now becomes 16%. Part B wants me to find the percentage of the population that is heterozygous, or 2PQ, for the same gene. So that would be this section that I've worked out, and the answer is 48%. Okay, so 2 done. Let's look at question 3. Guys, if at any point you need, I'm moving too fast, or you need the video paused, just ask. Okay, question 3 starts off with, in a population of 100 students, 25 exhibit the recessive phenotype. Okay, as soon as you see the words recessive phenotype, your brain needs to clue in that they're talking about Q squared. So Q squared in this example is equal to 25 out of 100. And yes, I know most of you probably just did, 0.25, and that's fine, but there are going to be times where that number, that total population, isn't 100. Okay, so let's do the same thing. So if Q squared is 0.25, then, oh my goodness, then Q is going to be 0.5, because we square rooted 0.25. P is going to be 1 minus 0.5 to give us 0.5. P squared is going to be 0.5 times 0.5 to give us 0.25. And 2PQ is going to be 2 times 0.5 times 0.5 to give us 0.5. Okay, so now let's look at the question. All right, so now that we've found the parts and pieces, let's look at the question. So part A of this question wants to know how many students show the dominant phenotype. So this question takes us in a completely different direction than what we've been thinking about up until now. There are some things you're going to have to remember. When they use terms like dominant phenotype, they want to know what trait is being shown. What characteristic is the dominant characteristic that will be shown? So if you think about it, there are two ways for you to have the dominant phenotype. If we go back to that wavy hair example, if you were big W, you had wavy hair. Or if you were big W, you also had wavy hair. Well, big W in Hardy Weinberg is represented by P squared, whereas big W is represented by 2PQ. So the first step in solving part A is we're going to have to figure out what is the total frequency for P squared and for 2PQ. So let's go back and get my trusty green highlighter, and let's find P squared, and here is 2PQ. Well, 0.5 and 0.25, that gives you 0.75. Okay? But the question asked how many students. So not what was the frequency or what was the percentage, but how many students. So you would then take that 0.75 and you'd multiply it by your total population, which was 100, and the result is going to be 75 students as your answer. Okay, that's A. Let's look at B. B wants to know how many students are just homozygous dominant. Sorry guys, hold on a second. Right, sorry. So I was saying, so for part B, they want to know how many students are homozygous dominant. So this time, we only care about the p-squared value, but again, they're asking about students. So not just the percentage, not just the frequency, but the actual number of students. So it would be 0.25 times 100, because remember, 100 is how many are in our original population, and that would give us 25 students. Okay, part C wants to know how many of these students are heterozygous. So now we go back and we look at our 2PQ number. So this one right here, we've already done this one. So that's our 0.5 value, but if they still just want to know student count, so it would be 0.5 times 100 to give you 50 students. Now, one of the ways you can double check that you're doing things correctly is you can add things up to make sure that the numbers are equaling 100. Remember, on this part A, you can't factor that in because this is dominant phenotype. This isn't just dominant allele. Okay, so now let's flip over and let's jump to question 5. Okay, so again, we're jumping to question 5. So in question 5, it tells you in a population of 2,000 students, 180 exhibit the recessive phenotype. So we have a q-squared value. Q-squared in this scenario is 180 over 2,000, which is the same as 0.09. So now let's go through and solve all parts. So if q-squared is 0.09, it means that q is going to be 0.3, which means that p is going to be 1 minus 0.3, which is 0.7, which means that p squared is going to be 0.7 times 0.7, which is 0.49, and 2pq is going to be 2 times 0.3 times 0.7, which would be 0.42. Okay? So part A of this question wants to know how many students show the dominant phenotype. So again, just like part A of the question we did not too long ago, if we're talking about dominant phenotype, we need to think about who's going to be homozygous dominant and who's going to be heterozygous. So that's our 2PQ, and that's also our p squared. So we would start by combining those. This question I haven't actually solved. I'm just doing it from my head. So I got to do the numbers for this one. Okay, so that should be, if we did 0.42 plus 0.49, the result would be 0.91. But since it asked how many students, it has to be 0.91 times the 2,000 that's in the original population to give us 1,820 students as our answer. Kind of like writing in this highlighter or marker or whatever it is. Okay, so let's look at B. B wants to know how many students are going to be homozygous dominant. So again, we're only looking at the p-squared value. The p-squared value in this scenario is 0.49. So to get students, it'll be 0.49 times our 2,000, which would give us 980 students. And then our C Question wants to know how many students are heterozygous. So we look at the 2PQ number. So 2PQ in this scenario is 0.42 times 2,000 to give us 840 students. Okay, and if you wanted to do a quick double check, you could do 980 plus, you could do 980 plus 840 plus 180, and it should all add up to 2,000. Okay, so that's one of the ways you can double check. All right, so at this point, we've done, 4 examples of what this could look like and how this could appear. So you've had some practice. So what I'd like you to do with the time that you have left, I gave you a handout called Hardy-Weinberg Problems 2014. Okay, it should be front and back. I think it's 15 questions total. I would like you to do all the odd numbered questions for this. Okay, so on the Hardy-Weinberg problems, okay, 2014, you're doing all the odd numbers. Okay, so I'm not real sure what time it is right now, but you should work right up to the bell. Okay, if you don't finish all of them, you can bring it back to class on Thursday. But the expectation is once this video tunes out, you will work on those problems. It's not that you're just going to sit there, okay? All right, so real quick before I go, let's make sure that we kind of are aware of where we should be for the day. So we've done all of our agenda. We've done our genetics recap that was kind of built in. You've practiced some Hardy-Weinberg with me, and now I've assigned you some of those problems to try on your own. Okay? Pay attention to where it is you feel that you're having a problem. Where are you struggling? Where is the breakdown? So that when I come back, we can address that. Don't forget you have a phylogenies video that you need to watch tonight. All right, guys, have a great day and a good rest of week, and you will hear from me on Thursday. Bye.